All right, everyone, welcome to Kara's thesis defense. I think we all know why we're here today. A few logistics to begin with. We'll have our public defense now, at which any of you, family members especially welcome, can ask questions. And then we will retreat to my lab with Kara's thesis committee. We'll have a meeting, probably last an hour or so. We expect that to go well. We would never let a student stand up here and present if we didn't anticipate a very enthusiastic and positive outcome. And then if any of you are around 1 o'clock today, you can come on back to lab. There may be libations of various kinds available in the lab. So that's the plan. So a little bit about Kara. I promised no embarrassing slides, no embarrassing stories, so I'll play it pretty straight, but still say a few things that I think are important for me to tell Kara at this time. So Kara, as you probably well know, came to us from Wellesley, from Dave Ellerby's lab there. She already had a very strong research background at that time, not just on fish, but well, quite a variety of other things, plants and invertebrates. Gasp. But soon here, she found her way to the fish department, and where she's happily, hopefully, remained ever since. I think Kara's produced a very diverse and interesting thesis that she'll, of course, be telling you about in a few minutes. There are 4 things that come to mind when I think of Kara's time here in the lab. The first is she's brought outstanding quantitative skills, and not just in terms of calculation ability, but just thinking quantitatively about biological problems in general, and about fish and fish diversity and fish locomotion in particular. I think it's been a much needed infusion into the field. I've learned a lot from her as she's sort of pulled me along on this journey, so given my limited challenges in that department. The second area where I think Kara has made substantial contributions, really more unique in my lab recently as we've become more biorobotic, is in the quantitative comparative phylogenetic methods area. Again, I'm not even be able to really to be pulled along in this state, but I've learned quite a bit, and Kara really is just really our paper as one of her thesis chapters in Journal Evolution. She had a beautiful cover with a series of x-rays. We'll see some of those images in the talk from her work doing really, which material she really learned how to do this on her own and did a fantastic analysis for that evolution paper. So that's been really a new aspect of the lab, and I know others in the lab are sort of working along that vein too, but Kara really has done a premier job with that. I think the last 2 things I want to say about Kara are particularly personal to me, I would say. First is her love of the fish collection. Really a second home for Kara. And I felt the same way when I was a graduate student, with some of the same staff members when I was a grad student. It's a tremendous resource. I think Kara has used it so well from a scientific point of view. She's used it well from a personal point of view, getting involved in the collection, helping with the collection, understanding the value of the collection, helping undergraduates with work in the collection. It's really been a great source of pride for me to see how intensely she's used the collection and developed a tremendous love for the collection. And I think the last thing I would just say about Kara, which again hits me a little personally, is her love of the history of the field, her love of a desire to understand what's come before, going back into the 1800s. This has, of course, led her to haunt the MCC library, looking for cast-offs of important historical books. We have representatives of the MCC library here who have been very important in providing Kara with an incredible library of books, which will have to be moved at some point. But it's been great to see how much she's embraced the history of the field and gotten pleasure from the amazing work that's come before that's often forgotten in these days when if you can't get a PDF of it, well, forget that. So I really appreciate that personally. I've loved having you in the lab. I've learned so much from Kara. I'm thrilled to be able to stand here today and to tell you that she's going to present the results of her PhD thesis. better.
Thank you also for the things I've liked most about being in the department. So that was really wonderful. I'm really excited to be here today and tell you about the work I've been doing to kind of untangle how body shape and morphology and kinematics, or the motion of how fish move, evolved and changed to support some performance. Really, it's been a lot of fun to work on, and I hope you enjoy the talk. So before I ruin all of your beliefs about me, people will say that I'm a fish biologist. I don't actually consider myself a fish biologist, nor, as some will claim, a cichlid biologist, which makes me cringe. I consider myself a biologist based on questions. And the question that motivates me the most is how organismal traits evolve to support performance. I'm really interested in this trait-performance relationship. And probably the best way to understand this was explained by Stephen Arnold in a classic paper in the 80s. He says you have morphology. That within morphology included a whole diversity of traits. So you have things like form, you have physiology, you have behavior, a whole suite of traits. These in turn affect performance in the field. And it's performance that's the subject of selection that eventually leads to fitness and the evolution of these traits again. So there's a feedback loop. And I'm particularly interested in how the evolution of these traits relates to performance given those constraints of common ancestry. So it's entirely possible that your ancestral fish wasn't as good in your current environment than you are now, and you're burdened with kind of that historical set of traits to begin with. Now, we're going to ignore fitness for this talk. That's a geneticist problem. And we're mostly going to ignore physiology too. And we're going to focus on how body form and motion, in this case kinematics, affect locomotor performance. Now from this diagram, you probably think that this is simple. This makes it look like this is a soluble problem. This is not a soluble problem. No. Comparative evolutionary biology and comparative evolutionary biomechanics in particular are a tremendous Gordian knot. You have the poor researchers, myself, George, many of you, trying to untangle a web of traits and how they relate to performance, function, what have you, back in evolution. They're all tied. And unlike Alexander the Great in legend, we can't just take a sword and cut the thing in half that defeats the purpose. So we have to kind of tease apart all of these confounding variables. Now I choose to do this in fishes. Why? Well, for one reason, fishes are incredibly diverse. It makes them fun to work with. Among vertebrates, they're certainly the most diverse. There are more than 30,000 species of fishes. They come in all different shapes and sizes and behaviors and environments. So they're a really interesting group to work on. Second, we have a kind of convenient pre-established hypothesis for how body form and performance are related to one another. This was mostly codified by Paul Webb in the 80s. It's been developed long since then. And we're going to run through it a little bit because it's basically the foundation for all my entire dissertation. So we have these three ecomorphological specialists, and then we have generalists in the middle. What do I mean by ecomorphological specialists? Each of these body shapes, each of these fishes, is supposed to be exceptionally good, an archetype of its kind, at a locomotor specialization. So here we have a tuna, and the tuna, it's got this fusiform body, deep in the front, narrow at the back, lunate tail. And this morphology is supposed to be the archetype for economical cruising. It's supposed to minimize drag while keeping thrust reasonably good, so you can move through the water steadily for a long period of time without using a lot of energy. Here we have a pike, which is considered the archetypal accelerator. It's got a big tubular muscular body, a lot of thin surface area at the back. And this guy is supposed to be our specialist accelerator. It's going to generate thrust with its muscles and its fins. It doesn't really care how much energy it spends doing that. It just wants to shoot out of the box. And in this corner, we have a butterfly fish, which is considered our archetypal maneuverer. It's got a deep, laterally compressed body and fins that are pretty big. And the idea is that this can generate turning moments to maneuver through complicated environments. So anything in between these three archetypes is some sort of a generalist. Now, the trick with this is that it is indeed a hypothesis. It is based on some observations of convergent evolution, but it really hasn't been rigorously tested. And unfortunately, the literature has kind of taken this hypothesis and run with it, to the point that if you see a slightly narrower peduncle on a stickleback, you say, oh, this is obviously adapted for cruising. We don't have the data to say that that subtle of a morphological change does indicate an adaptation for performance. So I really want to get at the heart of this hypothesis and see does it pan out in fishes. Now, like I said, we're going to focus on this form-performance relationship. But what ended up being the central question of my dissertation is how the hell do we even answer this question? George and I have had many conversations at his desk and say, okay, you want to isolate the effect of form on performance. How are you going to do it? It's really hard. So I took 4 kind of different approaches to answering this question, 4 ways of trying to tease apart shape and performance. The first, as I like to think of this as the nuclear option, where you remove all of the biological confounding variables, make a simplified model, and say, all right, how do these shapes perform? The second is an opposite perspective, where I say, obviously biological variation matters. Let's look at all of the evolved variation in the lineage and see how morphologically that fits in with our hypotheses. Then, having had some understanding of morphological evolution, I said, okay, how can we actually separate morphology and our kinematics, the motion? And finally, I started to think about more than just steady swimming and the biological relevance of that and how we might measure it aside from form. And we're going to just dive right into chapter 1. And I'm going to start by mentioning the specific hypothesis that I wanted to test in this chapter, namely that this narrow caudal peduncle and the lunate tail of a tuna shape enhance performance. Now, the evidence for this is largely based on models and convergent evolution. This shape is shown up multiple times in fishes. It's shown up in reptiles. It's shown up in mammals. It's happened again and again and again. Probably that means it's useful for some hydrodynamic function. So instead of using fishes, which have confounding variables, I made a bunch of pieces of plastic. I cut them out in the shapes of fishes to test the hypotheses of narrow caudal peduncles versus deep and forked versus unforked. And I made them in 3 different stiffnesses because I had a sneaking suspicion that stiffness and shape might interact to change performance. Little did I know my future lab mate, Kelsey Lucas, was working on this during her undergrad at the same time and probably doing a better job. I tried. So I attached these pieces of plastic to what is now perhaps infamously known as the Lauder Lab flapper. It's a device that moves the foils back and forth in heave. It also does pitch. I only looked at heave. It measures the forces and torques on the foil. And this whole apparatus is inside basically a fish treadmill, a flume. So there's a current running past. The foil moves the water behind it, which you can image to look at hydrodynamics. And we can measure the forces and torques being produced at the same time. Now, there are a number of ways this could have worked out. Presumably, so the darkness of the points here is going to represent shape, and the color is going to represent stiffness. Presumably, there would be some relationship between performance and the different shapes. Perhaps that relationship would be consistent with stiffness too, and then we can interpret it. I don't really care how stiffness affects the shapes. I'm interested in the shapes, so that's all right. And I don't really care what the pattern across the shapes is. I can figure it out as long as it's consistent. This is what I got. None of those things. So this is self-propelled speed. It's kind of how well a foil is doing given a particular motion program, how fast that foil can go. It's one metric of performance. I measured several. Don't bother staring at these too much, but there's forces here, there's power consumption, there's energy, there's torque. What I can tell you is there were complicated interactions between shape and stiffness for every single one. Every one. And let me tell you, it is not terribly satisfying to write a paper where your conclusion is this is really complicated. Now, at this point, you might be thinking, well, that's a wash. And admittedly, I probably did. But that wasn't all that I had. I also had hydrodynamics. And for a moment, I want to focus on the single foil, which is the coral foil 1. It has a narrow peduncle and a forked tail. had reasonably decent performance in all of my measures. And this is the flow across the foil at self-propelling speed at this plane here. So where you got the trailing edge of the body and the leading edge of the tail. And I want you to focus on this blue vortex. And this is coming off the trailing edge of the body. If we jump forward in time, we see that vortex has moved and is interacting with the flow at the leading edge of the tail, so here, to generate what we call in biomechanics a leading edge vortex. Now, leading edge vortices are really important in biomechanics. They enhance lift while reducing drag. And because this is a lift-based swimming device, as most fishes are, or many fishes are, I shouldn't say most, that probably does actually improve energy economy. That's very exciting. And in fact, leading edge vortices have been observed, have been hypothesized in fishes for a long time. They've been models using computational fluid dynamics. They've been observed in things like moths and swallows. And it's the same mechanism that makes our delta wing planes, like a Blackbird or a Concorde SST, really, really good. So this was really exciting. But there's one thing I noticed when I was looking at these, which kind of saved the study in my eyes in many respects, which is that if you don't have this trailing edge and a gap in front of the tail, you don't get a leading edge vortex. So this suggested to me that you need predictable patterns of morphological variation and evolution and co-variation of structures in order to reap a hydrodynamic benefit. Does shape actually evolve that way in fishes? So obviously the next step was to go look at fishes and see, okay, do these shapes, do the body and the fins evolve in correlated patterns as I expect? One way to think about this is, I like to say, is a fish like Mr. Potato? So we see these fishes, they look very aesthetically pleasing to us. It's kind of what we'd expect fishes like this to have in terms of fins. Can we just plug and play? Swap the fins out. Maybe fins occur in cassettes where you get a whole suite of fins at once, but the body shape doesn't really matter. So I wanted to really investigate this question of how body shape and fin shape might evolve together or independently. We didn't know. Most studies had only focused on the body and ignored the fins, despite their importance. Now I'd like to, because as George mentioned, I really love history, use this as a chance to bring up my favorite physiologist, August Crowe, and mention Crowe's principle, which is that for a large number of problems, there will be some animal of choice, or a few such animals, for which it may not be most conveniently studied. Through a variety of factors, I came across such a system in cichlids. Cichlids are an adaptive radiation, explosive radiation of fishes, distributed across the neotropics. You have species in Sri Lanka and India. You have species coming out of Lake Tanganyika, radiating into the African Rift Lakes and the Congo River. And then you have a whole neotropical lineage. And as you can tell, by looking at the crazy diversity of cichlids, you see they have the morphological variation that I need to really test this hypothesis. And they have an added benefit of that we know their phylogeny really, really well. So I went to my beloved fish collection. I said, give me all your cichlids. And I took radiographs of some 131 genera. And I marked the body and the fins. So in the red, you see geometric, morphometric landmarks for the body. You see dorsal fin measures, caudal fin measures, anal fin measures. And I did principal components analysis and partial least squares canonical analysis to look at the variation within structures and the variation between structures, respectively. This is just a small sample of the many radiographs I took. I've never been more grateful for a lead-lined radiography room in my life. So, as I said, I'm interested in evolutionary variation in these structures. It's not enough to just measure the fish. I need to account for their ancestral pattern of diversification. And this should be a no-brainer in cichlids because, as I said, their topology, the topology of their phylogeny is very well understood. We know the relationships between cichlids. If you look at the colors here, which denote lineages, you can see there's agreement between these two trees. So this is from Friedman et al. 2013, and McMahon et al. 2013, in terms of where the clades are relative to one another. I needed to use two phylogenetic hypotheses because the cichlid world is a bit controversial. And if you look at these two trees, which were published at the same time, They're very similar, but they differ in one major feature, the branch lengths. So if we were to look at the timing of events in these two trees, we see that this tree says South American and African cichlids split about 46 million years ago. And in this tree, they split about 60 million years ago, 70 million years ago. And because the analyses that I'm using are very sensitive to branch length, and because I didn't want to pick a side, all of the analyses I'm about to show you I did 3 times. Once without any phylogenetic information on the whole data set, and once each for each phylogeny using the subset of my data that corresponded with that tree. I'm only going to show you the non-phylogenetic data, but take my word and the word of 27 pages of supplemental materials that the phylogenetic analyses and the non agree completely. They're almost identical. So instead of showing you the principal components analysis plots, I'm just going to give you kind of the results of those because, again, individual variation isn't as interesting to me as correlated variation. But this gives you a sense of what was important, what varied in these fishes. So if you look at body shape, about 1/3 of the variation in body shape was explained by body depth. People will say this is elongation. The same pattern's been found in lots of other lineages of fishes. I prefer to say body depth. In the dorsal fin, the major axis of variation was explained by either uneven distribution of fin area, with either most of the fin area at the back, or very even distribution of fin area. The pattern in dorsal and anal fins was the same. This is going to be a recurring theme. The dorsal and anal fins often do the same thing. The first major axis of variation in the caudal fin was length with respect to body size. So what this is showing is either you have a very long fin given your body size, or even a very short fin given your body size. And finally, the last axis of caudal fin variation is explained by fin concavity, where you have either a forked tail or a convex tail. Now, if we think back to our hypotheses of what matters in fish evolution, then we see, okay, this looks good. It seems like the structures that are interesting and that we hypothesize are important are varying in fish. Great. But how do they covary? So this is a partial least squares canonical analysis plot. What this analysis does is it picks up pairs of canonical variants that each individually explain a lot of the variation in their block of data and also correlate with one another. So we're picking out the patterns of maximum correlated variation. And again, because I did all these analyses also with phylogeny and accounted for, means like this correlated evolution as well. So here we see the major relationship between body and caudal fin shape is that deep round bodies come with long fins. This is good. Long, narrow bodies, short fins. Okay. When we look at the pairwise relationship between median fins, we see that kind of even median fins vary across from kind of long fins. They go together. Dorsal and anal fins tend to do the same thing. Good. When we look at the dorsal fin and the anal fin relative to the body, we see again narrow bodies going with short fins, deep bodies going with long fins in both cases. And if we look at this other plot, which shows a slightly less strong relationship than the others, this is looking at body shape and caudal fin shape. We see what this looks like. How do we describe this? The loadings showed that caudal peduncle elongation, so essentially elongation and tapering of that region before the tail, co-occurred with a forked tail or a semilunate tail, whereas the more truncated body happened with a more concave tail. Convex. I'm never going to get that right. Now, again, these are relationships that were plucked out from the data. I didn't superimpose these particular features as important. So it's good that the individually varying features are also what's showing up in the maximum covariate patterns. Now, if we look at all of these pairwise relationships together, and we squint a little, and you say, what does this mean in terms of global covariation? We end up with kind of these 3 extreme forms. And when we look at these 3 extreme forms with respect to our hypotheses for what ecomorphological variation looks like, we get very excited. So it looks like cichlids are actually, have evolved, showing correlated evolution that corresponds with our purported ecomorphological specialists. This is great if you're a Paul Webb fan, which I am. That's not enough. It's not enough to show that things evolve their morphology in a way expected with our locomotor hypotheses. We need to show that they actually perform as expected. They might do totally different things. We don't know that our steady swimming morphology actually steadily swims well at all. So I decided to do a simple 3-species comparison to solve this problem, fix fish morphology forever. I took 3 cichlids. I'm calling this our archetypal cruiser. This is a cichla. We have an archetypal maneuverer. This is Physidon. And archetypal accelerator, kinematics. These colors are going to be used throughout. And at this point, I'd like to take a brief moment for a methodological note. These trials that I'm going to show you involved swimming a fish with gradually increasing speed, usually for about 3.5 hours per trial. I had to increase the speed by 0.2 lengths per second every 15 minutes. I had to watch the fish consistently to see if it fatigued. So I was very grateful that lab mates could step in when I needed to leave the room for some reason. But you'll also notice in the following slides, there isn't a lot of data for this fish. And that's because it didn't swim. I tried everything. I tried flashing a strobe light at it. I tried hitting the tank. I tried covering the tank so I couldn't see anything and it was in the dark. I tried, and I'm not making this up, cutting the hook off of a fishing fly, hiding in the corner, casting into the flume, and seeing if it would go for it. Because the only thing that makes these things move is food, and that didn't work. So the data you do see from this fish were taken with something hiding it from me. So I can't tell you how deep it was, which means it might have been on the floor of the tank. So take every piece of data from this redfish with a giant grain of salt. That being said, I think it says something about the selection biomechanics have had for really only studying fish that swim well. So there's a lot of data on trout, there's a lot of data on bluegill, and these are fish that you can get to swim steadily well. It might be that most fish don't swim steadily well. So even though I selected these species not for good swimming but for morphology, there's probably a reason you may want to think about including more species that don't swim well. Okay, that being said, they performed as expected. Our cruising fish, cichla, had a higher maximum sustainable swimming speed. So it could go for a long 4 hours, 5 hours without stopping at fairly high speeds. And that was significantly different from the only other fish that we have good data for. Great, I solved it. Fish performed as expected. Swimming morphology is done forever. No. So I can't tell you that those differences in performance were due to shape. Because what you see here is the midline kinematics of these fishes, all swimming at the same speed. And you might notice, if you take a critical eye, that the fish that swam the best is doing something very different than the fishes that did not swim the best. So the motion pattern might actually be what's driving the difference in performance. I can't tell you that shape is what's doing that. Not to be set off, I say, all right. Here, if you look at the fins, they're also doing different things. So you have lots of different motion patterns that are confounding our effective shape. How do we deal with this knot? We need a way to separate the motion from the form. In other words, we need a way to quantify gait. Gait, again, is the pattern of steady motion that you use when you're moving at a steady speed. So you might be familiar with petrotop gaits. Before we can even get to performance. Now, we have categorical designations for fish gaits. So we have a variety of body caudal fin swimmers. They're usually named, the type of swimming is usually named after the kind of fish and how much of its body is moving. We have oscillatory body caudal fin swimmers. We have median impaired fin swimmers, and these include pectoral fin swimmers that undulate their fins. They include pectoral fin swimmers that oscillate their fins, dorsal fin swimmers, anal fin swimmers, and both median fin swimmers, both undulatory and oscillatory. Now this is good for understanding what changes across fish gaits, but you can't compare across categories. And there's certainly no way in this categorization to account for fishes that use some combination of these, because some fish have a transition between labriform pectoral fin swimming and caudal fin swimming. Other fishes might use all of their fins all of the time. So there's not a lot of flexibility to quantify variation within this framework. Again, because I like history, I went back and I said, where can I figure out how to do this? And there's a very good model in my mind in the work of Milton Hildebrand on tetrapod gates. He really revolutionized the field of tetrapod biomechanics by coming up with a way to plot tetrapod gates. So here are a bunch of symmetrical course gates. He found that you could parameterize gait in terms of, in this case, duty factor and phase lag. Doesn't really matter for our case. You can plot them, you can compare them. So here, for instance, is a walking gait. Here's a trot. And here's a really strange gait called a passo. So it doesn't really matter what they are for my purposes. What matters is that the idea is you can find quantifiable parameters that you can then use to compare things analytically. So how do we do this for fish? Well, first we know that they use different propulsors. They can use their pectoral fins, they can use their caudal fins, they can use their bodies. We might have to account for the different use of propulsors. Often we'll measure fin beat frequencies as a way of parameterizing gait. So here you don't have to look at the data too much. These are caudal fin beat and pectoral fin beat frequencies for the 3 species of fish that I've studied. But my point here is that we measure this. We do it all the time. You can measure fin beat amplitudes. So again, how far your fin is moving, that's probably important too. So mind you, this is very hard, and this data, I should add, was mostly digitized by me, partially digitized by Brianna Lang in undergrad, and I'm really, really grateful for her help. But finding where the end of a translucent fin is very difficult, and again, I'm really excited to see what Kelsey Lucas does with this in a couple of years, because I never want to have to do this again. So we have these kind of parameters that we might use when we're parameterizing fish gait. So how can we think about graphing a fish gait then? This is the framework I came up with. If you consider locomotion and you consider gait as an output that is a product of multiple propulsive inputs, so caudal fin input, dorsal fin input, pectoral fin input, what have you, you can define some input function that characterizes what that propulsor is doing. I like to use something I call effort. I know it's a bad name according to some, but you can all deal with it. I thought of it first. And effort we're just going to call, actually Paul Webb thought of it first, but he didn't name it. And I thought it frequency times amplitude. This is something you can pretty reliably measure for all of these propulsors. Then you can choose your output of interest. This can be often speed, it could be acceleration, it could be, once we have the capacity to measure it, again, I'm excited to see where Kelsey Lucas goes with her work, because I think we'll be able to measure it someday. You choose an output. Then you can consider each one like an axis, your propulsors and your output. And again, these aren't going to be independent. We don't care. We're not looking for independence, we're looking for a quantitative description of gait. And in fact, the relationships among these variables is what's going to be important. If you do that for my 3 species of fish, you get a cloud of points that looks like this. This is a gait space. I'm very excited. Now we're going to remove our redfish because it doesn't swim. And if you plot a trajectory through gait space, this shows the combination of propulsive movements that these 2 species use at any given speed on the z-axis. And if you look, you can already tell they're separate. And because we don't think in 3D, I'm going to break this down in 2D. So if we look at this plane here, we see that pectoral effort, so the input of the pectoral fins from a discus, increases with speed. The input of the pectoral fins from cycla really doesn't. If we look at it from this point of view, we see that the caudal fin effort of cycla increases with speed. But for discus, it doesn't until the very top speeds. And if we look at it from the top down, we get an analog of Hildebrand's gait space. We see, okay, this is the combination of pectoral movements and caudal movements that this fish uses at all speeds. And you can tell from this, these fish are doing very different things, but we've quantified how different they are. And that's what I wanted to do. So this is very exciting, and hypothetically, you can plot any swimming fish on this space. So we have a new comparative tool for studying fish gaits that lets us go where tetrapods have been for a long time, but where fish biomechanics have dared to tread. So now that we have our fancy technique for disentangling form and motion, I've solved the problem. I can link form and performance, right? Obviously. No. We have to remember that this is in fact inside a giant Gordian knot of traits. And the one trait I haven't really discussed yet is behavior. As it turns out, fishes don't usually swim steadily. So I want you to pay attention to this jack. He'll come around again. Oh, look, he flicked his tail. Oh, he did it again. Look at that. And if you spend most of your time watching fishes, you'll see that that's the norm. And though routine swimming is criminally understudied, the one study that did do it came to the same conclusion. Routine swimming was dominated by unsteady motions of linear, and he said centripetal, angular acceleration. Routine swimming was not quasi-steady. So we have a problem. We can understand steady swimming really well, or we can start to think about behaviors that might be more biologically relevant. Well, I love challenges. And given our hypotheses, perhaps it's not surprising to know that fishes don't usually swim steadily. Why would we expect an acceleration specialist to swim steadily? Why would we expect a maneuvering specialist to swim steadily? I probably should have realized this before I set off. Though I will add that understanding steady locomotion, it turns out, and probably from the beginning, is very important when you want to generalize further to unsteady. So liking a challenge, I said, all right, I want to do for steady gaits what I want to do the same for unsteady motion. So here I've got basically a gait space like I described to you before, but instead of different propulsors, I have different aspects of caudal fin beat locomotion. Start with one propulsor, then get more complicated. Here we have caudal fin beat period, here we have caudal fin beat amplitude, and this then is going to be some representative fish's hypothesized steady swimming gait trajectory. An unsteady movement then is a movement between two speeds. So how can a fish do that? It can move, maybe it iterates through all the intermediate kinematics between those two speeds. Maybe it decides to use a couple of fin beats of a very short period before drifting back into its new speed steady kinematics. Maybe you switches to a larger amplitude and then goes back into its steady kinematic. But the point here is that we can think about unsteady motions as a path through the propulsive space that's not always at the same speed. So this was the idea I had in my head when I set about to solve the problem of unsteady motion. And because you don't want to use fishes that don't swim to do something terribly complicated, I was very fortunate that Valentina DeSanto took a bunch of videos of unsteady motions of bass and trout that she let me use to kind of test out my methodological strategies. I'm going to focus on trout just because of time. So here's a steadily swimming trout, again in a fish treadmill that's going to look like it's not really moving. Here's a linear acceleration, so you'll see this fish gradually moves forward. That means it's increasing speed. And here's a burst acceleration. It's a bit more chaotic. I apologize for the GIFs, but putting a lot of high-speed videos is hard. So we have these three behaviors. They're going to be encoded by these three shape forms. I said, all right, the first thing we might do is we might discretize these heterogeneous behaviors in terms of fin beats. So if we look at individual fin beats, right? And if we look at individual fin beats with respect to maximum instantaneous acceleration, you might notice that the overwhelming pattern here is color. Color for these points is the initial speed. So in fact, the kinematics of your fin beats are really driven more by initial speed than any unsteady thing going on, which is very frustrating if you're me. But if you just look at the bursts, which are the stars, you see they're often doing something different. So yes, there are differences across acceleratory behaviors. But even if you try and zoom in on this variation of non-bursts, it's the same. They're all intermixed, different shapes, so the colors are really what's dictating the fin beat kinematics. So looking at fin beats in toto, not a good way of really understanding what's going on in unsteady locomotion. So then I said, okay, maybe that's the wrong approach. maybe I want to think about this in terms of steadiness itself. So what I did here was I saw what's the most steady thing I can think of? A perfect sine wave. A perfect sine wave is perfectly steady. So I fit the 4 parameters of a sine function to the data for each of these trials. This is detended tail tip motion. And I said, how well is the tail tip motion represented by a sine wave? And I did the correlation between the best fit sine wave and the actual data. And this was pretty good. So here we have a steady swimming. You see the correlation with the sine wave is 0.93. It's not bad. And I'll admit that this is actually low for steady swimming. Most of them were actually 0.99, which is ridiculous. If you look at linear acceleration, we have a correlation of 0.71. Again, not bad. Also, this is low for linear accelerations. They were usually typically correlated with their sine waves by more than 0.9. And bursts are doing something totally different. They're just very inconsistent. This one has a correlation of 0.53 with its best fit sine wave. And while this isn't a perfect method, for one thing, the correlation coefficient isn't, the behavior is not linear with your variation from your sine wave. That's a bit peculiar. This is only going to be as good as your optimization algorithm for fitting. There are limitations, but if I were going to derive some machine learning approach to separate behaviors, this is probably what I'd attack. But it's not perfect. I said, okay, what if we try what I initially thought of, look at the fin beats through propulsive space relative to, say, instantaneous speed. So this is, here we have the blue points documented by numbers, so that's the order they're in, and black points are steady swimming fin beats at the same speed. All right, so there's not much going on. This is a steady swimming trial, so it's the same. This, ignore the offset, which is very small and due to a slightly different initial speed. But again, we see the fin beats in fin beat space are in the same place with steady speed for the linear acceleration. There's no difference in linear acceleration tail beat kinematics between that and steady swimming speed. And the bursts are doing something strange, but there are so few fin beats you can't generalize. So again, imperfect, but something. I also thought, hey, let's look at tail tip net acceleration and compare that to the acceleration of the body moving forward. So here we have the blue line, which is the tail tip. We have the orange line, which is body acceleration. And sometimes they're correlated, and sometimes they're not. And it turns out the trials that I picked to show you actually are better correlated with each other than usual. Isn't that super reliable? And it's very disheartening. So once again, I find myself with a chapter that kind of ends in, well, this is complicated. But I made an attempt to kind of parameterize these unsteady behaviors, and I think this is work that people can build on. So yes, we can do this. We're going to need either a lot more fish, or sophisticated methods, or honestly, probably both, before we can kind of do for unsteady motions what I've made it possible to do for steady state. So where does this leave us? Well, I use simple models to show that while shape and stiffness and shape and performance interactions are incredibly complicated, you might need correlated patterns of shape variation to reap any hydrodynamic benefit. I looked at the diversity of cichlid evolution to show that shapes actually do exhibit correlated evolution in the patterns that we hypothesized at the beginning. I looked at steady swimming and was able to come up with a method to quantitatively describe gait, which we can then use to study comparative studies of gait and shape and performance. And I'm really excited to use this in the future to do all the cool questions that I really want to answer. And I started thinking about how we might parameterize unsteady locomotion so that we can start addressing more and more biologically relevant behaviors. With that, I hope you'll all forgive me for a moment before I answer questions by thanking some people. Certainly my committee, George, we've had so many fascinating conversations, mostly about history, but also other things. You've let me have more independence than perhaps a grad student ever should. But thank you for letting me try to answer my crazy questions and in some cases succeed. Andy, you've always given really good advice, and I think you know your biomechanics more than anyone I've ever worked with ever. So I really appreciate your input, and you've always had thoughtful things to say. John couldn't be here today. He's always encouraging and kind of helped me think about adaptive relations, funding sources. I've gotten money from people and used it to buy fish and fish food. I need to give special thanks to my undergrads, Alex Whitehart, who is my first thesis student, who's right here, and Brianna Lang, who, again, she's helped me with my fish care and digitizing. She's been a better assistant than anyone could ask for. In the science world, there are a few people who deserve special mention. My undergrad advisor, the George Lincoln Dave Ellerby, who has grown into the best mentor and friend I could ever want. And he stuck with me for the rest of his life. He couldn't be here today, but certainly he shaped the way I think about science more than any other individual. Dave Coughlin, who is also become a mentor, I actually went to him when my pike cichlids wouldn't swim, and I said, hey, Dave, is it possible these don't have any slow oxidative muscle fibers? Is it possible physiologically my pike cichlids can't swim and I'm not a failure? P.S. He went and did this for me. They do have slow oxidative muscle fibers, the jerks. I guess they could swim if they wanted to. Lots more research to do there on behavior of swimming. All of these people, and I'm sure some whom I've forgotten, have given me help with methods or just useful discussions and thinking. And I've kind of adopted so many people into what I think of as my scientific family. These are all big sisters and brothers to me. And I just really appreciate all of you. And no one could that be said more than for my cohort. I had a lot of trouble thinking about what to say about you guys. I'm really not good at being eloquent when it comes to acknowledgements. You know who you are, you all know how much you've done for me, and you probably don't know how much you've done for me. I would stick out my neck for you any day of the week, and I value you guys more than anything else at Harvard, including the fish collection. It hurt a little bit, I'm sorry. But I don't think you guys have any idea how much I appreciate you, and I wish you did. No two men have helped me more in my pursuit of finishing my dissertation than Karsten Hertel and Andy Williston. I don't know where Karsten is. Is he okay? But Andy's here, and yeah, they'll say, oh, we just moved heavy jars for you. And yes, you moved heavy jars and opened stuck jars, but I've gotten more unexpected ideas and discussions and friendship and so much from both of you. And I don't know what I'm going to do without you two when I'm not at Harvard. Expect emails? Yeah. Maggie is the glue that keeps the water lab together and the glue that keeps me together. And she has had to listen to more bull excrement than any other person with respect to the completion of this degree. I couldn't have done it without you, and that's not an understatement. The Ernst Mayr Library has become my third home away from home. Mary Sears is here and Ronnie Broadfoot. I see them all the time. We have philosophical discussions. We talk about old books. I poured through ancient things for fun more than I care to mention. And I couldn't do it without their letting me have my free reign in the stacks. So I really appreciate them as well. And also they can find manuscripts from the nether reaches of some cyclopean hell. I don't know how you do it, but you find manuscripts everywhere. Water Lab, you've been amazing, past and present, all of you, lab mates. I have no words. You know how I feel about you guys, right? And finally, these three people. My mom, who's this one, if you can tell, has been the single most important person in my entire life. She's encouraged my thoughts in science when I liked science, and even for that brief period when I didn't at the end of high school. She always encourages me to do whatever I can, and she'll say that I'm brave and she's not. And she is full of it. Because the only way I can do all the things I do is because I have her telling me to go do it. And I love her. This is my brother. He couldn't be here today. I love him. He encourages me. He's the smarter one in the family, so I always have something to strive to. There are a few things I'm better at him than. I hope public speaking is one of them. Love people. And this guy, this is Ryan, who somehow, though he started dating me during my qualifying exam, kept dating me, even though it was during quals. And he's kept me supported and happy and loved and surprisingly well fed for the past 4.5 years. So I probably would have died of malnutrition had he not stepped in. And with that, I'll take any questions.
Very nice. I'm going to let Kara handle the questions for a few minutes here, but I'll just add, because I know you said you don't consider yourself a fish biologist or even a cichlid biologist. But for those of you who may not know, there's a long and extremely distinguished history of work on cichlid fishes here at Harvard, going back many years.
Yes.
So Kara's following in a very distinguished set of footsteps. They were all very famous. So you have a little path ahead of you here. Yeah.
I should add that one of the things George is hinting at, and many of the specimens that I actually used were collected in the 1860s during the Feilich expedition by one of the Agassiz, literally out of Harvard. So people who've been working on it was year long.
Carl Lehman Melnis asked me a fantastic history of cichlid work, which Kara is well represented in modern science. Questions for Kara?
Yeah, Jack.
It's amazing how much you've been able to quantify these sort of like somewhat at first maybe hand-wavy ideas. Do you think that sort of for moving forward, is there a way to quantify the process of deciding what things are performance? Or is that something where sort of intuition is going to be always sort of?
I think in many respects, our ability to quantify that would be limited, but to find a data-driven way of thinking what performance is would be very difficult. The best way I could think about doing it is seeing what correlates with fitness with respect to routine behaviors. Measure everything during a routine scenario, do the population genetics to see what matters, and then go back. That seems like an inhuman amount of work. That being said, I'm largely of the opinion that if you think about it for long enough and scribble on enough pieces of paper, you can eventually find a way to quantify almost anything. But that's just kind of a philosophy I've adopted. So I think there might be better ways of thinking about what counts as performance. I'm not sure how you quantify the decision behind what is performance, but that's a really good question.
Yeah. Given the complicating factors in chapter 4, would you, if you could reconsider the pyramid to, instead of being the 3 extremists and the generalists, would you reconsider the pyramid and perhaps make it pentagonal or add a section into it?
So I'll tell you what I would do. I'd go back to my cichlid morphospace. I would take morphospace. I would measure the performance and say those 3 things. In all of them, I'd see what the extremes were in any direction, and then I'd decide what shape to use based on that. I think the fact that the triangle, I mean, Paul Webb is no slouch, and every single piece of work I've presented is, I think there are maybe at least a page and a half in my dissertation of references for Paul Webb. So I think it's a really good starting point, but I think we have the ability now as a manpower in fish biomechanics to have a data-driven set of what extremes and performance and morphology we have. Yeah, Kadeem.
So, so you didn't talk as much about the generalists as the 3 specialists, so it just occurred to me now whether, let's ask, with the generalists, do you see that they're either just trending towards an extremer of some kind, or is it some sort of balancing selection?
I imagine they'd be trending. I mean, if I were to show you Paul Webb's version of that diagram, there are fishes at intermediate places in different locations on the triangle. I only like to speak confidently about things I've measured. And as I've only measured those 3 species, and I've never measured a quote-unquote generalist in the different aspects of performance that I looked at, I'm not comfortable saying, yes, they all lean this way or that. We have no idea what they do. We haven't measured enough of their performance. That's largely the problem with biomechanics, is we know about beluga, we know about trout. The rest is a big black box that hopefully will keep me employed. Yeah, Martin?
Yeah, so in your phylogenetic analysis, so where you have the two trees, you said your analysis is very dependent on the branch length of those two, and then you didn't show like the details of your follow-up.
Yeah.
Do those results in any way allow you to make assumptions about which one of those two truths is closer to the actual truth? Or is that not something that you can like completely
I
understand?
have a data-driven opinion on which of those two phylogenetic hypotheses is more true. If I were to say it, I would immediately alienate myself from half of the sickness. Yeah,
Supplemental materials.
No, they're just in the supplemental materials of my brain. And if you can find it far enough back, a hot Twitter conversation with a member of the opposing party.
All right, well, Kara, thank you again.